Hi, welcome everybody. We are going through week 2 content. I really enjoyed meeting you all on Tuesday. Those of you whom I have not met, I look forward to seeing you in the future. As I mentioned in class on Tuesday, week two's content is basic biology. We sort of have to slog through it. Um, I'm trying to keep things to a reasonable need to know level, but we'll just kind of introduce the topic, talk about some genetics and genomic concepts. We might as well get started. Okay, so what's the difference between genetics and genomics? When I first introduced to my family that I was teaching this course, my husband said, "Well, I know what genetics is. What's genomics?" Well, the difference between genetics and genomics is subtle. A lot of people use them interchangeably, but genetics is your basic science, your pure science of the mechanisms of heredity and the variation of inherited traits. Genomics is broader. It's more applied science. It's the study of the entirety of the function of a genetic sequence, including interactions among the different genes and interactions between genes and environmental factors. It includes therapeutics. It includes anything that has to do with the genome. Why do you need to learn about genomics? Why am I teaching a required course in the curriculum on nursing for genomics or genomics in nursing? Well, because many diseases and factors that prevent disease have their origins in your genome. Science is finding all kinds of ways to diagnose, treat, and prevent illness based on our evolving understanding of the human genome, and these are becoming increasingly available to healthcare consumers. Nurses are in the position to help people understand their options related to genomics, to manage their health conditions, um, mitigate their risks, but we don't typically and there is a lot of literature on this, have the knowledge to do it effectively. And so, agencies that accredit schools of nursing have identified genomics as an area that needs to be addressed. Makes sense. And Gwinnett Mercy, in response to these standards, has decided that it will be required for you to take some coursework in genomics in order to graduate. And I know that that is important to you. So we'll continue. Brief history of genetics. One of those sort of you have to do it before you get out of it. We all know Darwin, survival of the fittest, evolution of a species. He proposed that traits can be more or less adaptive to the environment, and people will, or species will selectively mate based on the fittest individuals, so people who are most adapted to that environment. And so you get that survival of the fittest. Mendel conducted his experiments in the garden with some peas and showed blending of genetic material, traits, and patterns of inheritance. And that's where we get like our Punnett squares and things like that from. Miescher was a scientist who discovered that the material inside the nucleus of a cell was composed of nucleic acids, and that was the preliminary evidence of DNA. We finally knew, okay, we knew that there were traits that were passed down, but how does the body regulate that? How does that happen? Discovering nucleic acids sort of paved the way for, maybe that's where the cell houses that material. And then molecular biologists were able to see chromosomes inside the nucleus of a cell. They saw those nucleic acids arranged in those nice little X-shaped or Y-shaped bundles, as the case may be, using staining and a standard microscope. So those were kind of important things that happened. And then scientists later discovered that that material did contain the genetic code responsible for inheritance. Watson and Crick used the work of previous scientists to propose the double helix structure of DNA, that twisted ladder that we all see. And all of this sort of contributes to our understanding of the raw material that is responsible for inheritance. Biological scientists were able to induce mutation in genes using gene cloning and things like that. So we saw that the environment could, in fact, induce mutations, most of them not adaptive, some of them a little more adaptive. From all of this scientific work, people decided that because inheritance governed so many things, they saw social characteristics that were passed down in families, they came up with a theory that selective breeding could encourage evolution of a better human, more intelligent, higher in character. It began as early as the early 1900s, where we started to see involuntary sterilization of people housed in psychiatric facilities. It became really famous in Nazi Germany during World War II, when experiments were performed on twins, horrific ones. And then it continued really in subtler forms on smaller scale, um, even as recently as the 70s, and maybe even further, but where we involuntarily sterilized massive numbers of Native American women based on social characteristics, with no deference at all to the social determinants of health or the conditions that they were living with that predisposed some of those things. Um, but that's eugenics, and that's kind of important because it influences our cultural attitudes. It could influence policy making when it comes to things like the CRISPR project, where we are directly editing genes. And it gets into that ethical debate. What eugenics really was, it was thinly disguised ableism and racism that looked like science to a lot of people. We know that those characteristics are influenced by many things other than genetics, but there are still people who think that, you can build a better human by allowing certain people to breed and not others, or by editing certain genes and leaving others alone. So I'm kind of giving that a mention. You might see it again. Okay, so in 1990, we see the beginning of the Human Genome Project, which was a massive undertaking. Think about it. I know I mentioned in class, one of my classes, that if you wanted to build the human genome out of Lego bricks, you would need more than 10 million of them. So that was not a project I was interested in doing in class because, Lego bricks are not cheap. But it is a huge, huge undertaking. There's so much genetic material in the human genome. So in 2004, they actually completed the project and published their work, which was really exciting. And now they're able to sort of work with it. Contemporary genomics, we have some trends and issues. We alluded to them in class, but I'll repeat them here. Genetic testing is becoming much more available and it is much more extensive than it used to be. Even paternity testing is much more precise. We used to try and prove or disprove paternity based on blood type, but it was sort of a probability of whether this person could in fact be the individual's father. Could not say for sure whether the individual was that person's father. Now we can look at DNA. We can use it for all kinds of things. It becomes a little sticky when we talk about things like accessibility and affordability. For example, NIPS testing, non-invasive prenatal screening, can isolate fragments of fetal DNA in the maternal bloodstream. It is non-invasive, so it can be offered to people. It's not going to risk their pregnancy like CVS or amniocentesis does. However, it's not covered by insurance. So if we're saying that it's important to offer this testing, if this becomes a standard of care, how can we deny it to some people and give them a lesser standard of care? Similarly, we have BRCA testing. It could be useful information to help women make decisions about prophylactic mastectomy or oophorectomy, even increased screenings, increased mammograms, increased ultrasound, increased screening for ovarian cancer, which we don't do universally. If we're going to deny that to some people by making it unaffordable, then we have sort of an ethical issue there. Now, a lot of these products you may have seen, 23andMe, Ancestry.com, MyHeritage, I think is another one, where they direct market genetic testing to consumers. A lot of it is for entertainment purposes only. You find out your ancestry by ethnic and racial characteristics of the DNA. It'll tell you where your ancestors were from. It can tell you who you're related to. You might not have ever known you had a fifth cousin in the UK, and now you do. But the most sort of shady part of that direct marketing genetic testing would be where they give you a readout or printout for an extra charge, of course, on the risk factors for various diseases. Without expert interpretation, there's no way to know how this information is being used by consumers? Are they sharing it with their physicians? And is the testing even accurate? There's no real oversight for these companies. And are they marketing your DNA to other people? Yeah, there's all kinds of stuff that goes on with that direct marketing. Genetic testing can detect a lot of inherited disorders. The ones that predict risk are probably the least utilized, but maybe the most helpful in the end. So if that can be perfected, if we know that somebody is at risk for diabetes, we can target that person for a more frequent testing. We can get them hooked into programs that prevent them from progressing to type 2 diabetes. That could be important in the future. So it might be something you need to know. Therapeutics, we have the CRISPR project. I know I alluded to that in class, where we directly edit genes responsible for causing diseases. And that is exciting technology. It's where some of the ethical considerations come from. People are afraid that we're going to use that technology again for that eugenic aspect. And then we have therapeutic cloning, where you can clone tissue or cells. You can even clone, if you don't have stem cells, because they are hard to come by, and there's an ethical component to that. We'll discuss it later. You can take skin cells and turn them into liver cells if you do it right. Because all cells have a copy of that basic script. So that is an area where you may see some changes. And then we have pharmacotherapeutics as probably the most widely used now, and it's really in its infancy. But you see products like Genomind, where they do some genetic testing, and based on variations for the CYP450 enzymes responsible for metabolizing your medications, they can identify which medications are most likely to be effective and which ones are least likely to cause side effects. Now, if you can find medications for somebody without all that trial and error that are both safe and effective, you can really address the roots of their disease a lot more efficiently. So those are just some contemporary genomics and issues. And that concludes our first video.